Good afternoon, and welcome to the first in a series of 5 webinars, which are organized in the next few weeks to commemorate the end of the PREPARE project, but also to celebrate a new beginning inspired by PREPARE. My name is Menno de Jong. I'm a clinical microbiologist at the Amsterdam University Medical Centers. I'm deputy coordinator of PREPARE and also co-PI of the MERMAIDS study, which is the topic of today's webinar. But first of all, I would like to briefly introduce PREPARE for those who are not familiar with the project. Can I have the next slide? So PREPARE is a large-scale and also ambitious project which started in 2014, is generously funded by the European Commission. And the aim of the project was to establish a collaborative network that facilitates harmonized clinical research on infectious diseases across Europe and is ready to rapidly respond to severe outbreaks with relevant clinical studies and thereby providing timely evidence to improve clinical care already in the course of the epidemic and also to inform public health responses. Obviously, PREPARE has been put to the test during the current COVID pandemic. And as you will hear during current webinar and the future webinars the next few weeks, PREPARE has done so very successfully. The foundation of PREPARE is a large network of hospitals, of primary care sites, and of laboratories throughout Europe. And in this network, during the project, several large observational and interventional studies have been performed and are still ongoing. And these include the ALICE study, which is an adaptive study on influenza treatment in primary care. It includes the REMAP-CAP study, which is also an ambitious adaptive trial, but in the ICU setting, and the observational MERMAIDS study in hospitals and primary care. And all of these studies will be discussed in the course of the coming webinars. And today we will start with presenting the MERMAIDS study. And before we begin, just a final remark. After the talks, there is ample time for questions and discussions. So if you have a question during any talk, please post this in the chat box. I will collect the questions and then moderate an as lively as possible virtual discussion after the talks have completed. So without further ado, it's my great pleasure to introduce the first speaker, Professor Peter Horby, who will introduce the MERMAIDS study. Please go ahead.
Great, thank you very much, Menno. My name is Peter Horby. I'm Professor of Emerging Infectious Diseases at the University of Oxford, and I'm the PI of the MERMAIDS studies. So MERMAIDS is the multi-centre European study of major infectious diseases syndromes. Not a bad acronym. Next slide, please. I'll just give you backgrounds on one of the MERMAIDS studies. Next slide, please. Because there are actually 3 MERMAIDS studies within the portfolio of these observational studies. So there's one on acute respiratory infections, one on arboviruses, which will be the subject of another seminar. It's arboviruses in the Balkan regions of the European Union. And one on pediatric infectious diseases, which includes an RE component. So today you'll hear about MERMAIDS RE, and also the RE component of the pediatric MERMAIDS study. Next slide, please. So the MERMAIDS RE, these studies have both the scientific objective, but also an organizational objective. One of the organizational objectives is to set up, a platform for responding to outbreaks with clinical research. And as you'll see later, we did pivot this MERMAIDS RE platform to COVID, and that's had multiple benefits, as you'll hear later. So the primary objective of the MERMAIDS RE study, which we started several years ago, was to identify host and pathogen-related determinants of severity of community-acquired acute respiratory infections in adults, really to try and understand the pathogenic processes, the mechanisms as to why some people get very sick and other people don't. We know at a very broad level there are risk factors for severe disease, such as comorbidities, obesity, age, etc. But it doesn't really help us understand the underlying pathogenic mechanisms, which we want to understand so that we can target interventions at those pathways, but also so that we can better identify who is at risk of deteriorating and ending up with severe disease, so that we could target measures early. So that's the prime objective. The secondary objective really was to look across the sites in Europe at the etiology, clinical management and outcomes of patients with acute respiratory infection, so that we could have an understanding of the heterogeneity of treatment practices and patient mix across the network, which would allow us to really design studies with a better understanding of the heterogeneity in the network. And then also to look at prognostic and diagnostic algorithms related to the biological measures that we're, biological parameters that we're measuring. So the plan was a prospective observational study of 500 adults. You'll hear more about the pediatric component later, in primary and secondary care, and across multiple countries and primary and secondary care sites in Europe. Next slide, please. So to answer the primary objective, we want to look at differentially expressed genes as expressed by the RNA transcriptome. So there have been these transcriptomic studies done, but at the outset we undertook a review of those, and we saw that actually most of them were too small to be able to answer key questions about pathogenesis and risk groups, because either they were too small to stratify by either disease or patient comorbidity, or they were just looking at one patient subgroup, like those in intensive care. So there was difficult to have a comparator group looking at the transcriptomic profiles. So how it's designed is basically to develop subgroups of patients. Those with mild and severe disease is the first level stratification, to understand why some people have severe disease and some people have mild disease. But then stratifying by the infecting organism. So here we've got, you know, some key pathogens: influenza, rhinovirus, respiratory syncytial virus, and by the presence or absence of different comorbidities. Then we could directly compare the transcriptomic profiles in, for example, as shown here, somebody with influenza and no comorbidity. We could compare those groups with severe disease and those with mild disease, and see that once you control for the comorbidity and the infecting pathogen, do you see a different transcriptomic profile, which may give us indicators of pathogenic pathways. Next slide, please. And so the analysis really is looking at differentially exposed host genes, stratified by pathogen and comorbidity. You'll see here some of the pathogens and the comorbidities. And so next we'll have James will take us through the site setup and the recruitment, and that will be followed by Louise talking about a description of the cohort that was enrolled, and then later by Cosimo, who will give you the primary preliminary results of the transcriptomic analysis. So over to you, James.
Good afternoon, everybody. My name is James Lee. I'm also at the University of Oxford, and I've had the delight of managing this project for the last 5 or 6 years. So I'm just going to briefly show to you how we've set up the study, and just what the study numbers and the sites and countries that we've recruited from. So I'm just going to briefly show you the inclusion criteria for the study. So we recruited from primary care, so GP practices and hospitals. So these are our mild cases recruited from primary care, and then the more severe cases that are hospitalized. We recruited adults over 18 years old. They needed to have a clinical suspicion of a new episode of an acute respiratory infection within the last 7 days. So it was vital that we captured patients within the acute phase of respiratory infection. They needed to have a temperature and also a respiratory symptom and systemic symptoms. So patients presenting with a temperature, a cough and a sore throat and a headache, for example. These were our cohort that we were looking for. So we recruited over a 28-day period, and it was a serial sampling study. So we take samples on day 0. So for patients that are admitted in day 0, we take a sample, and on day 2, For the ones that are in hospital, we also take a sample when they're discharged, and then all patients are followed again on day 28. And those samples consist of nasopharyngeal swab sampling and a series of blood samples. And this is where we recruited from. So mostly all across Europe. We recruited in 8 countries, 38 sites in total prior to COVID reactivation. We recruited from 9 primary care sites and 29 hospitals. And this is what recruitment looked like throughout the duration of the study prior to COVID. So we were set up in 2016, and things started quite slowly because we had to set up contracts with the sites and just getting people up and running. And then in 2016, we were raring to go and we got the sites all up and running. Season 3, as you can see there, we recruited a lot more. So if you may recall, in 2017, 2018, that winter period was when we had increased cases of influenza B across Europe. So that actually resulted in a lot more hospital admissions and GP admissions. And then our final season in 2018, 2019, we also recruited. So that brought us to a total of about 1,500 patients from 2016 to 2019. And that gave us about a 2-to-1 ratio of hospital patients to patients recruited from primary care. So this will give us good comparator groups for the analyses that Cosimo will be showing later. And this is how we recruited per country. So as you can see there, the majority of the recruitment come from the UK, but we recruited well from all of the countries that we were set up in. So yeah, this has worked really well, and it's a good representation of a European study and what we can do in terms of having partner institutions set up across Europe. So I'm not sure if Louise is online to present some of the clinical data, so I'm going to
I'm here, James.
Oh, you're there. Sorry, Louise. Oh, thank you. So yeah, Louise, you'd like to introduce yourself and present the data.
Thank you, James. So I'm just going to briefly present some characteristics of the PREPARE MERMAIDS cohort study on patients that were included. Next slide, please. So I'm going to present the baseline characteristics, the cohort characteristics of the inclusion. So this is, as James said, the patients that were included in the MERMAIDS RA study from 2016 to 2019. So the pre-COVID era. So this is a cohort of patients presenting in primary care or secondary care with symptoms of acute respiratory infections. And we had a fantastic recruitment. Thanks to all the sites, it's been a great collaborative effort, and thanks to all the patients who signed up to take part in the studies. So looking at the baseline data, we can see there's slightly, the secondary care cohort of about 1,000 patients is slightly older. The median age is 60 compared to 44 years old in the primary care cohort. There's a slightly higher proportion of men in the secondary care cohort, but it's not a significant difference. It's predominantly, ethnicity-wise, predominantly white ethnicities included. And looking at the days from onset of illness to inclusion or into the study, the median time from onset of illness inclusion is around 3 to 4 days, so no difference between the primary care or secondary care cohort. We find a higher, not surprisingly, slightly higher proportion of people who are smokers or ex-smokers in the secondary care cohort. And there's also a slightly higher proportion of people who had a recent influenza vaccination within the last 6 months in the primary care cohort compared to the secondary care cohort. And we also see there's a higher proportion of people with multiple comorbidities, so 2 or more comorbidities in the secondary care cohort. So next slide, please. So looking at comorbidities, the dark blue bars shows the proportion of different types of main groups of comorbidities in the secondary care cohort. The light blue bars represent the primary care cohort. So we can see that chronic cardiovascular disease is the highest, most common comorbidity, followed by chronic pulmonary disease, and then metabolic diseases where type 2 diabetes is the predominant one. Next slide, please. So symptoms, looking at comparing the symptoms on inclusion. So as James mentioned earlier, to be included, the patients had to have a self-reported fever within the last 7 days or a measured temperature of 38 degrees or higher, and a couple more of the symptoms, RE symptoms or systemic symptoms. So there's no main differences in symptoms except for a higher proportion of patients in the primary care cohort documented with sore throat or runny congested nose compared to in secondary care. And we also see a higher proportion of people with dyspnea in the secondary care cohort. Next slide, please. So we looked at there's a lot of data in, this is preliminary data from the analysis. It's just to show an example of some of the assessments and some of the severity scores that we have included parameters for in the case report forms. So not surprisingly, the pulmonary severity index, for example, the median is higher, significantly higher in the secondary cohort compared to primary cohort, and also the other symptoms such as a high respiratory rate and a pulse above 100, and there's a high proportion of people with an O2 sat below 95% in the secondary care cohort. And we've also, looking at the data, as we expected, as a lower proportion of records on, for example, biochemical samples and also diagnostics in the primary care cohort. We can see a significantly higher level of CRP above 10 in the secondary care cohort. Next slide, please. So this shows the median length of stay of hospital stay in the different countries. So overall, the median length of stay was 5 days. And we see some variation, significant differences between the countries, which might indicate a potential heterogeneity in the inclusion criteria. In the inclusions, sorry. Next slide, please. So at the end, so the samples were at the end of each season, all the patient samples were batch shipped to our reference laboratory at Antwerp University for diagnostics with RT-PCR. And this slide shows the viral diagnostics carried out in the reference lab on all the samples. So of the viral diagnostics, the most common pathogen detected was influenza, which was detected in about a quarter of the patients, 25% of the patients in both cohorts. So of the patients with influenza, the influenza B was the most common type detected in the primary care cohort, 54% compared to 30% in the secondary care cohort. And influenza A H1N1 was the predominant type in the secondary care cohort, in the hospitalized cohort, compared to the primary care cohort. So for the bacterial diagnostics, we've looked at the local diagnostics performed by each site. And as you can see on the slides here, there was very limited number of patients that had been tested and had a bacterial pathogen recorded in the case report forms. So secondary care, there was only about 1/10 of the patients that had bacterial pathogen detected locally. And of those, strep pneumonia was the most prevalent pathogen, followed by Legionella. So looking at, so as I said, it's preliminary data. So looking at the antibiotics, we saw that 63% had been administered antibiotic on inclusion in secondary care, but there was a limited records on bacterial diagnosed and a lot of missing data to draw any conclusions. Next slide, please. So looking at the outcomes, about 94% were diagnosed with an acute respiratory infection locally at outcome. And luckily, a majority of patients survived. There was no case fatalities or mortalities at day 28 in the primary care cohort, and 1.4% mortality only in the secondary care cohort. And we can see that of the people admitted to hospital, about half received supplemental oxygen and 4% was admitted to ICU. And only less than 1% received invasive ventilation. So next slide, please.
Thanks, Louise.
Yeah, so in summary, influenza was the most prevalent pathogen detected in about 25% of patients, and the outcomes, and there was a very low case fatality rates at day 28, which was great. And I'm now going to hand over to James to talk about mermaids in the COVID era.
Yeah, thank you. Yeah, so just to put into context, so all of that clinical data that Louise has just gone through, all of that was data collected up until the beginning of 2019. So, a good year before the COVID-19 pandemic hit the world. So with mermaids, we were able to reactivate for mermaids for COVID-19. And this was all due to being set up as part of PREPARE. So PREPARE has got different outbreak response modes. And in January, we were able to prepare what we needed to do across the networks in order to mobilize ready for a research response. And because the funding had come to an end, we were able to actually apply for additional funding from the European Commission to use the fantastic networks that we built up across Europe to respond to COVID-19. And this response was called RECOVER. So this is the Rapid European COVID-19 Emergency Response. So we were able to reactivate mermaids and recruit patients again. So this just gives you a quick overview of the timeline for PREPARE and then that additional funding we received at the start of 2020. And this is really good. So because we had PREPARE already set up and we had contracts in place with sites and they were all familiar with how to recruit patients with acute respiratory infections, it means we were able to quickly respond for COVID-19. So what we did was we increased the recruitment target and extended the study to February 2022. And we're uniquely positioned to answer questions for COVID-19. So we're able to compare those mild and severe cases of COVID and compare them with other respiratory viruses that we've detected in previous seasons, so influenza, for example. We can compare those mild and severe cases and stratify by comorbidity. And importantly, as with our primary objective, we want to look at the host responses and pathophysiology in COVID and look at those different mechanisms that underlie disease severity. And we already have some preliminary data from the last year, and Cosimo, in the slides coming in a few moments, is going to present some of that really interesting gene expression data. So I showed the recruitment prior to COVID-19, and as you can see here, we were able to rapidly respond and recruit patients because of the pandemic and because of this pre-established network of sites set up as part of PREPARE. So not only is this going to be able to give us further insights into COVID and what we understand about the disease, this really shows the importance of having pre-established networks and sites set up ready to recruit into research and observational studies. So before I hand you over to Cosimo, I just want to briefly thank a lot of the sites. There is a lot more people that have recruited patients into the study than these guys, but this is just some of them across Europe. And yeah, thank you to all of the networks and sites involved in recruiting patients, because without people on the ground dedicated to recruiting into the study, we really wouldn't have the success that we can show today. So now I'm going to hand you over to Cosimo, based at the Amsterdam Medical Center. If you've got any questions, please wait until the end, and yeah, we look forward to answering any questions you may have.
Good afternoon, everybody. Thank you for joining this afternoon session. So in this presentation, as James already introduced, we are going to have a look at how gene expression profiling could help us to understand or gain insights into pathophysiology to acute respiratory infections. And also, we're going to try to explain why mermaids, in this sense, represent an important knowledge base for this type of studies and future studies on this topic. So next slide, please. I would like to start with an introduction, with an overview of what this talk is about. And I will first provide an overview of study design and objectives. And I will briefly introduce what transcriptome is and how we try to reduce complexity of this type of data and steps needed to achieve this reduced complexity. And I will then present results on host gene expression in COVID-19, and then followed by an overview of how COVID-19 sits into this into the context of other acute respiratory infections, and we will discuss in the end results and future plans. Next slide, please. So on the design and the objectives, we are. I will start with the COVID-19 data. It is a currently a major public health concern, and for this type of data, we included 109 laboratory confirmed cases that were matched with an equal number of healthy controls, and the matching was performed based on age and gender. And it's important to mention that this is one of the strengths of MERMAIDS, and this is a clear example where we were able to include these healthy controls because they were actually the day 28 or recovering or convalescent samples from the phase 1 MERMAIDS ARI. These 109 confirmed cases were separated into 3 distinct severity groups based on the type of care required by these patients, namely mild cases, at home patients at home isolation monitored by the Amsterdam Public Service, the health service, moderate cases, hospitalized patients, and severe cases, ICU or critical ill patients requiring ICU admission. And I would like to mention also the fact that we made sure that this classification was also matching the WHO criteria for severity in COVID-19. Next slide, please. We also included influenza and RSV infected patients or samples from MERMAIDS phase 1 in order to compare them with COVID-19, and we retrieved these samples from primary care and hospital care. So the final goal, well, the general, let's say, aim of this of this study was to collect whole blood to study gene expression using microarrays, and the idea behind this was to gain insights into COVID-19 pathophysiology, but also to identify molecular determinants of progression to severity, and also isolate dysregulated pathways that could serve as starting point to also identify predictors of the outcome and possible targets for treatments against this disease. Next slide, please. An overview of the patient's characteristic of this COVID-19 cohort. What we in general found, what we observed was that the numbers were in line with other major respiratory infections, and in general an increasing age and days of illness, percentage of any comorbidity, and percentage of death in more severe cases compared to mild and moderate. And it was interesting to notice that 86% of the cases were male in severe COVID, but this was also in line with what has been described in the literature. Next slide, please. If we want to link clinical information with gene expression, and that becomes really important to first reduce the complexity of this system. As I mentioned before, transcriptome, the transcriptome is a complex system. We have to imagine that we are dealing with, on average, 22,000 genes in a human cell. We have, of course, several cells, and these genes, we have to also keep in mind that these genes are involved in different pathways and in different interactions, which makes this system indeed quite complex. And it's really important to reduce this complexity in order to interpret, biologically and clinically interpret these results. So the step that usually the following step basically summarize the main pipeline to achieve this complexity reduction. So usually we start with global, we look at the global variation or perturbation in gene expression. This is done usually with principal component analysis or another approach called molecular distance to health. We are going to see this later. And after getting an idea of the global perturbation, we then proceed with trying to actually reduce this complexity using transcriptome module decomposition, and this is done because we can reduce such a complex system into small components or single components that explain certain biological function. Modules need to be seen as a group of biological and functionally related genes. We will see this as well later. Then after detecting modules, we can look into them and see which genes are differentially expressed. And differential expression, basically, could be summarized into, we want to compare how cases differ from controls. We do this usually with simple t-tests, and we measure the magnitude of this difference by fold change, as well as significance using of the difference using p-values. In the end, the final goal is usually identifying a bunch of genes that we prioritize, and this could be our candidates, biomarkers, or drug targets, or whatever our analytical, say, pipeline, our final goal it is. So next slide, please. All right, let's have a look at the actual data. This is the principal component analysis. In simple terms, this plot shows how single samples are separate from each other. We have reduced the space into two simple dimensions. And what you can see here, that the green dots represent healthy controls, and the other dots encapsulated into this solid circle represent COVID-19 cases. What we see is that in general, COVID-19 cases are separated from healthy controls. That was what we would expect. It was also interesting to see that within the COVID-19 group, there was a clear separation between mild cases and severe cases, with moderate cases being intermingled between these two groups. Okay, so if we go to the next slide, we can also see how, beside the principal component analysis, another interesting score, this time working on one single dimension, could be used to detect the global perturbation, score actually the global perturbation of transcriptional profile of each patient compared to healthy controls. This score is called molecular distance to health, and it has been it's a well-established approach. It looks at for each sample how this sample deviates from control. Again, what we can see is that mild cases are closer to healthy controls than moderate and severe cases, with severe cases being far away from control. And why it is important, this type of score is important. In the next slide, please, if we look at the next slide, we could actually see that this score can be used to associate clinical traits with global perturbation of gene expression. And this is done mainly because we would like to understand what are if there are main drivers in our clinical data or in our baseline information that drive the variability in our data. What we found is that age, as expected, seemed to be the most the main driving parameter or clinical trait in this data, as well as comorbidity, but not but we couldn't say the same for gender and illness duration. So there should be something else also driving variability in this data. So we looked into the genes, and if we go to the next slide, please. We looked into the genes that actually contribute the most to variation in our data. And we wanted to identify the modular architecture of this gene expression profile. As you can see in this heat map that shows controls on the left and SARS-CoV-2 cases on the right, based on these most perturbed genes, we were able to distinguish 3 different clusters based on severity, clusters of severity, but also within these clusters, we noticed that the patterns of expression were distinct across severity. So the idea is that, next slide, please. The idea is that if we naively try to split this profile into subcomponents, we should be able to detect what we called previously modules. And that's what we did. So we applied, and in the next slide, it's going to show that. We applied an approach that is called independent component analysis to dissect the gene expression profile into small independent components. And that what the aim of this analysis was to identify these modules, and again, which are groups of biological and functional related genes. And what we were able to do was identifying a number of modules that clearly represent specific immune system processes, as well as in immune system specific cell types. And in this plot, you can see this time, this is a plot similar to principal component analysis, but this time this was applied to single genes rather than samples. What you can see, the different colors basically represent different modules, and we were able to find specific modules associated to adaptive immune response, such as T cells, B cells, and plasma cells, modules associated to innate immunity, such as interferon response, myeloid leukocyte immunity, but also genes involved in coagulation and platelets degranulation, erythrocytes metabolism and development. And also a very interesting module that we were able to identify was the module associated to sensory perception of smell and taste. It was quite surprising to find this in these datasets, since we are working with the whole blood. But this finding may suggest that there should be some immune component driving one of the symptoms that are specific, or well, that have been described to be specific to COVID-19. And I'm talking about loss of taste and smell. So next slide, please. After identification of immune modules, we proceeded with identification also of differentially expressed genes within these modules. On the left side of this slide, you can see how these genes are shared between the different severity groups. What we noticed in general was that an increasing number of differentially expressed genes could be observed with increasing severity. This was in line and matched actually our molecular distance to health. But also we observed that most of the differential expressed genes were identified between hospitalized and ICU, and then a great number and a big number of genes seemed to be unique to hospitalized and ICU as well. But on the right side, and I think this is even more interesting, you can see how the genes detected in each separate module behave in terms of, enrich the list of differentially expressed genes. And this was really interesting because we noticed that interferon response seemed to be prominent in the first, in mild and moderate cases, and tend to diminish in severe cases, while plasma cells was present in all the three severity stages or three severity groups, suggesting that patients are probably developing memory response against this pathogen that they have never seen before. But what was also quite interesting was that the regulation of the immune response and regulation of myeloid cell immunity was increasing, as well as coagulation was increasing with increasing severity. On the other hand, concerning downregulated genes, we found an interesting and massive downregulation of erythrocytes, which seemed to be more prominent in moderate cases, but also was again interesting to see that sensory perception, the genes involved in sensory perception, were mainly downregulated in mild and moderate cases, but not in severe cases. And together with this, we also observed that genes involved in T cells regulation and T cells immunity tend to be more downregulated with increasing severity. Next slide, please. After identification of these modules, we wanted to validate and see whether they could indeed be associated in some way to clinical parameters. So we correlated the expression levels of these modules, or the scores, transcriptional scores of these modules with clinical parameters, or with parameters of severe infections. And we found that two of them, namely regulation of the immune response and CD8 T cells, CD8 and NK cells, were the models that mostly correlated with these parameters. But also quite interestingly, the same modules were the two that significantly associated with thrombotic complications in COVID-19. Next slide, please. So as mentioned in the introduction, we also wanted to compare COVID-19 to other respiratory pathogens, and in these MERMAIDS, again, it's one of the best studies to achieve this, given the richness of data available. So we compared COVID-19 with two major pathogens able to cause mild to the full spectrum of severity in acute respiratory infections. What we observe is that a great number of genes, differential expressed genes, were shared between these three pathogens, and this overlap was more prominent between COVID-19 and influenza. And with just few genes being specific to COVID-19, with a lot more genes being specific to influenza. So beside commonalities, we also wanted to look whether these small differences were significant or not. So we tested also differential gene expression between direct differential gene expression between these pathogens. Next slide, please. And this result and the results of this comparison could be summarized in this slide. What you see here, this slide depicts what we call volcano plots. On the y-axis, we have significance of differential gene expression. On the x-axis, we have the magnitude of the differential gene expression. So genes that tend to move towards or that are present on the left side, usually those are genes that are higher in other pathogens compared to COVID-19. Genes on the right side are genes that are highly expressed or more expressed in COVID-19 than in other pathogens. And what we can see, it's basically in general, we notice low expression, so lower expression of a sensory perception of smell and taste during COVID-19 compared to other pathogens. But also lower expression of interferon response and inflammatory and regulation of the inflammatory response in COVID-19 compared to other pathogens. And on the other hand, we saw a higher expression of plasma cells and immunoglobulins as we would expect in COVID-19 cases, together with higher expression of platelets and coagulation genes. So to conclude, if we go to the next slide, please. So to conclude, we have seen that we were able to confirm what has been already described in the literature, but also extends to using such a modular approach to look at the single immune components. We were able to see that severe COVID-19 is characterized by dysregulation, inflammatory pathways, and myeloid activation, adaptive immunity, so immune response in general, but also an increase in coagulation and platelets associated genes, and a decrease in sensory perception genes. We found also a considerable number of commonalities, together with few but relevant differences with other respiratory pathogens. And in order to understand whether these differences could be useful to better understand the pathophysiology and to gain more insights on the pathophysiology in the COVID-19, we are currently performing network analysis, because we would like to study how these modules interact with each other and how we could identify within this network candidate biomarker for prediction of outcome, and as well as identify drug targets that could be useful for to treat severe COVID-19. And there is a lot of work we are still planning, and it's still in our pipeline, because of course we want to make good use of the wealth of data available in MERMAIDS. This is an unprecedented study where we still have to analyze more than 2,000 patients with this disparate number of pathogens, and where we collected the full spectrum information about the full spectrum of severity, comorbidities, as it has been described in previous presentations. And then what the future plans are to gain more insights into the differences and commonalities, also in other respiratory pathogens. And expanding also the COVID-19 cohort in order to provide a global overview on how host gene expression could help with this in acute respiratory infection studies. Okay, in the next slide, I would like to thank all the people that were involved that are still currently involved in this study, especially colleagues from the Amsterdam UNC, Sarah, Karen, that helped us with the extraction of samples, Dirk, Hugo, Frank, René for the endless conversation about the data, Brandon and Menno for brainstorming and guiding me through the right analysis. Yeah, and also, I mean, colleagues from the Erasmus AMC in Rotterdam that provided us the ICU data, and Matthijs, especially Matthijs and Peter and Marion, and also, of course, colleagues from the Oxford University, James and Peter, and the GGD and funding as well. Thank you.
Thank you all very much for joining today and for staying on until the last talk. So as Cosimo said, I will be talking about the pediatric study in MERMAIDS, and the primary objective of the study, James, next slide, please, yes, was to estimate the proportions of children, of young children who were admitted to hospital attributable to different respiratory pathogens, mainly RSV and pneumococci, but also influenza virus and other viruses. And interestingly, we haven't got a lot of data on this from the post-conjugate vaccine era in Europe. We've got brilliant studies from low and middle income countries, but this MERMAID study is really the first one applying the same protocol for inclusions across a range of European countries. There were some secondary objectives. James, next slide, please. And I'm going to talk about two of them. One is that we also wanted to assess the association between co-detections of different pathogens and hospital admission. And the other one is that we wanted to characterize the management of children admitted for acute respiratory infections across Europe. Next slide, please. So recruitment was done continuously, so that means year-round through the acute respiratory infection season and through the summer. And recruitment started in September 2016 and concluded in March 2019. Cases and controls were recruited in parallel, and patients were recruited as they presented to participating emergency departments, and mainly during daytime hours. When you look at recruitment by month, you can see that, as you would probably expect, a large proportion of cases was recruited during the winter months, while recruitment into the control group was more evenly distributed among the year. And we recruited in 17 centers across 8 European countries. Next slide, please. Eligibility criteria for the cases group were mainly fever, and the primary reason for the hospital admission had to be a clinical decision that the patient was suffering from an acute respiratory infection, and there was a case definition provided as well. The next bit of the slide, please, James. So that was, in addition to fever, at least signs of respiratory dysfunction or of reduced general state, and they were able to be combined with clinically defined signs of upper and or lower respiratory infection. And children were excluded for having complex chronic comorbidities that would guide treatment, or for having a clearly non-infectious etiology of their admission or a non-RE focus. Controls were recruited from hospital outpatients or from patients attending for elective procedures. And what is important is that these controls did not necessarily have to be completely asymptomatic, but it was all right if they just did not fulfill the case definition for a case. So the study basically had two components. There is one case control part where we compared the cases and controls as explained before, but then the cases were also followed up during their hospital stay and until hospital discharge. And from all cases and controls on day 0, we took a nasopharyngeal swab, and that was analyzed for different pathogens. When we now look at the case control study population, you can see that there are some differences between the RE cases and the controls. The controls were on average 9 months older than the cases, and you can also see, as said before, that more cases were recruited during the RE season as compared to the controls. And there was also quite some difference between the different countries recruiting into the study, recruited more cases and some more controls. And all these differences meant that at a later stage during the analysis, we had to try and adjust for these differences as well as possible. Next slide, please. So for pathogen detection, which was done by multiplex PCR, what did we find? So the most prevalent potential pathogen that we found was clearly Streptococcus pneumoniae, and that was relatively closely followed by RSV and then multiple other viruses. What's interesting in the current situation is that about 5 to 10% of the children in the study had seasonal coronaviruses detected. And there were quite a few children with bacterial, viral, or viral co-infections. On average, you can, or in general, you can say that like the vast majority of children had some respiratory pathogens detected on these samples, and only in the control group there was a relevant proportion of children with no respiratory pathogen detected at all. When we look at the odds ratios for being a case upon detection of any of these pathogens, yes, please, James, the next bit of the slide, you can see that for a lot of these pathogens, it's actually very common to be detected in controls as well, and there is not much of an association between detection and hospital admission. So it really only is RSV and influenza virus, parainfluenza virus, and human metapneumovirus, where you can see a fairly good association between detection of these pathogens and association. It also looks like bacterial viral co-infection a bit towards the bottom of the figure. It looks like bacterial viral co-infection is associated with being a case, but when you look into the data in a bit more detail, the pattern of bacteria detected in these children is really pretty much similar between the case and control groups. So it more or less comes down to whether these children have the aforementioned viruses detected or not, and it probably doesn't really reflect true co-infection. As I said before, we had to adjust for a number of differences between the case and control groups. So on an adjusted logistic regression analysis, you can see that the odds ratios that I described before didn't really change much on the adjusted analysis. So we are pretty confident that these are fairly close to true measures of effects. I should mention that there were some additional sensitivity analyses done, and one of them was, for example, excluding any mildly symptomatic controls, and that resulted in a much higher odds ratio for detection of RSV and being a case. And this is basically what you would expect, and also one of the main reasons why we decided to have mildly symptomatic controls included in the study as controls, because we know that a large proportion of young children at any one time during the RE season have symptoms of an acute respiratory infection. And if you would exclude children with any symptoms of respiratory disease from the control group, then you would drastically overestimate the association between detection of pathogens and hospital admissions. And in the sensitivity analysis, we were basically able to show that this was also true for our study as well. So next slide, please, James. What does this mean when you encounter a patient at any time? So we looked at etiological fractions by age and season, and what becomes clear here is that there is quite a bit of seasonality to the contribution of different pathogens to the caseload that is seen in pediatric hospitalizations. And for RSV, for example, that meant that in the younger children, about half of the children who were hospitalized during the RE season were caused by RSV, while for influenza virus, the overall etiological fraction is lower, but there are groups like children who are older than 3 months who are hospitalized during the main peak of influenza season from January to March, and there influenza is quite a common pathogen as well. Okay, so next we move to the cohort of RE patients that we had in hospitals. So there were about 4 times more children with a lower respiratory tract infection among the hospitalized, and there is really a range of symptoms that children hospitalized with respiratory tract infections showed. What I would like to highlight is that among the lower respiratory tract infection children, quite a significant proportion in our study had signs of more severe disease. So about 2/3 of them, for example, had recessions, and about half, I apologize, this shifted a little bit here on the slides, but I can just say that this lowered oxygen saturation is the third set of columns from the right. So a bit more than half of the children who were admitted for lower respiratory tract infection had lowered oxygen saturations. When we go to the next slide, there are violent plots for the average white blood cell counts and average CRP values. And you can see that if you compare lower respiratory tract infections and upper respiratory tract infections, these values didn't really differ very much, and they also didn't differ between different pathogens. I'm showing RSV and pneumococci here. But what is interesting is that for CRP, there is quite a significant difference when you look at the rightmost plots. There is quite a difference between the children who received antibiotics and the ones who didn't receive antibiotics, which may partly be due to CRP guiding the decision to prescribe antibiotics to children. Next slide, please. When we look at management in more detail, we see, and I would like to focus on the lower respiratory tract infections here, we can see that about 2/3 of the hospitalized children received respiratory pathogen testing as part of their standard of care. About 70% had a chest x-ray done, and about 2/3 all in all received antibiotics. And in our study, a bit more than half needed supplementary oxygen. And we had about 5% of children who were admitted to an intensive care unit. And this is about a realistic resemblance of what you would also expect in like the real world scenario in a cohort of respiratory tract infection children. Next slide, please. Looking at the antibiotic treatments that children received, we can say that about 1/3 did not receive any antibiotics, about 1/3 of the children with lower respiratory tract infection, and among the antibiotics that children received, there was a bit of a difference between the different countries, but all in all, you can say that aminopenicillins, as would be suggested by current guidelines as well, were the most common class of antibiotics. And in some countries, aminopenicillins in combination with the beta-lactamase inhibitor were also quite common. Next slide. So looking at the outcome, first of all, I'd like to say that none of the children recruited in the study died. And again, this is something that you would expect because you would expect that in a high-cost country, about 1 in 1,000 children admitted for acute respiratory tract infection would die. So with about 360 cases enrolled, we wouldn't expect any deaths. The length of hospital stay was on average, or was a median length of 3 days, and the interquartile range was 2 to 5 days. And this differed quite a bit, probably as you would expect, between children with an upper respiratory and lower respiratory tract infection. Children with a lower respiratory tract infection tended to stay in hospital for longer. Next slide, please. Interestingly, when we look at whether children received antibiotic therapy or not, the children receiving antibiotics did stay in hospital for a longer time. And this may partly be due to the fact that the more severe cases would probably be prescribed antibiotics. But on the other hand, another explanation is that children may have stayed in hospital for longer to finish a course of intravenous antibiotics. Next slide, please. So what was interesting to us is whether it depended on the pathogens detected, whether children had to stay in hospital for longer. And there we can say that there wasn't really a difference between detection or non-detection of any of the pathogens that we looked for. So I'm showing these plots here for RSV and pneumococci, and there you can see that there is basically no difference. And this is also still true when you look at the rate of discharge adjusted for several different parameters that we collected at the same time. We also wanted to have a closer look at interactions of different pathogens and this outcome of length of stay. And there you can say that for a lot of pathogens, there was really no interaction, but we saw that having co-detection of influenza and RSV at the same time was quite strongly associated with having to stay in hospital longer. But this should be taken with some caution because this is a relatively rare phenomenon. And in the whole study, we only had 5 of these double positive cases. Next slide, please. Coming to the limitations of the study. So we probably had some bit of selection bias, and that was due to a convenience sampling approach. As I said in the beginning, it was mainly during daytime hours when sufficient staff was present at the different emergency departments. And because slightly different approaches to that might have been used at different study sites, it is quite difficult to compare study sites in pet mermaids. Another factor influencing or introducing some bit of selection bias was probably that as per the protocol, we requested some sampling, and that was upper respiratory tract samples and a blood sample as well. And this likely resulted in a sample that was biased towards sicker children, because children who were with relatively mild disease would, as per standard of care in many countries, not have any pathogen testing done and not have any bloods taken. The conclusions that we drew may also suffer a little bit from residual confounding, and this is because many study parameters that we collected are causally interlinked, and this makes it very difficult to adjust for all of them at the same time. So when you look at my previous slides and what we adjusted for in the different analyses, this is usually a fairly limited set of covariates, and this is because a lot of them would be causally interlinked. Another limitation is that the sample size with about 360 cases and about 320 controls was relatively small. The study was powered to see the difference between the different detected pathogens and an association with case status, but it was not powered for providing very narrow effect estimates, and therefore we have some wider estimates specifically for the etiological fraction estimates that I showed earlier. Next slide, please. There was some strength. It's the first European study that operated on this scale, and we've got a good geographical representation. It's important that we had year-round recruitment in the study, and thereby were able to capture seasonality. Then, as I elaborated before, representation of mildly symptomatic children in the control group is important to have realistic estimates of an association of a pathogen and hospitalization. Another strength is that we collected detailed clinical and management and symptom data. And what is actually something that generates a lot of richness of data is that we've got biological samples collected both from cases and controls, and are able to compare what we find in them. And then we've also got the gene expression samples also collected from children and controls. As Cosimo explained, mainly for the adult patients earlier, but analysis of the pediatric samples is still pending. So coming to the conclusions, you can say that there are a few viruses where detection of them on upper respiratory samples is strongly associated with RE hospitalization. This is mainly RSV, influenza virus, parainfluenza virus, and human metapneumovirus. but for a lot of other pathogens, detecting them on upper respiratory tract samples really doesn't tell you very much about the likelihood of being a case. About 2/3 of the children hospitalized for lower respiratory tract infections received antibiotic treatment, and we saw that length of stay was not associated with detected pathogens, but that children who received antibiotics, regardless of pathogen, basically had longer stays in hospital than those not receiving antibiotics. And with this, I would like to conclude and to say thank you to all the parents and participants who contributed to the study, and then to the staff at all the different study sites across Europe. And thank you very much for your attention.
First of all, I would like to thank all the speakers for their very interesting talks. We still have a little bit less than 15 minutes for discussions. I've received, in addition to several comments complimenting the speakers about quality of data and the talks, I've received a few questions. And before I will go over them, I would like to emphasize that questions still can be posted in the question box. So I've made a selection of the questions I received, and maybe the first questions are to James and or Louise. There were two related questions. One was whether in the MERMAIDS study there's an opportunity, or maybe you have already done that, looked at differences in clinical care approaches across Europe. And maybe related, are there differences in outcome between countries in Europe? Can you look at that? Is that something you're planning?
Louise, would you like to answer that one?
Thank you. Yes, so we have a big data set, and we have slightly delayed the analysis due to the COVID pandemic. So this is something we will look at, but it slightly depends on the like Malte mentioned, we have to be a little bit cautious around the potential different ways people have managed to include patients. Some sites might have been recruited more severe patients versus others, etc. So we'll look at the data, but also bear in mind that it could be affected by different ways they manage to include patients.
So it's planned?
Yeah.
To be continued. Then maybe a question. It said there are a few questions, and they're all also more or less related about dexamethasone, and maybe that's inspired by the presence of Peter, who, as you all know, has been the PI of this beautiful recovery study in the UK. So one of the questions is it possible, and that's maybe also to Cosimo, is it possible to understand better than we do now how dexamethasone actually works by looking maybe more into detail in gene expression profiles?
Shall I start, Min, and then hand over to Cosimo? Sure. You can give a more intelligent answer. I think it's interesting to understand. There's clearly an inflammatory process going on, and corticosteroids work through a whole variety of pathways. There's multiple targets. One thing we would like to do within the recovery trial, which we're not talking about now, is actually we have linkage to the Escherichia-4 cohort, so we can actually do genomics in a subset. So one interesting thing will be to look at genomics or even transcriptomics in those who respond or don't seem to respond to steroids. And whilst talking about the recovery trial, it's also exciting to see the sort of platelet coagulation signal in the MERMAIDS study, because we've got aspirin in the recovery trial, so it'll be really interesting to see if that figures there. But maybe Cosimo can take over now.
Unmute yourself, Cosimo.
Sorry. Yes, sure. I mean, definitely there is a link between looking to gene expression and drugs and way of action of certain drugs, and also that's going to be possible for dexamethasone. Yes, we have seen that indeed immune response signaling plays a major role in this in COVID-19, and I mean one interesting experiment, and also plays a major role in COVID-19, and there seems to be an association with thrombosis and coagulation. So it would be really nice to look into which type of pathways are perturbed during COVID-19 and after dexamethasone is given to these patients. Actually, this could also be tested in vitro. That could be really a really nice experiment, and then see how it compares with in vivo data, because we have a simplified setting there, and then see how it compares with the in vivo data. But the principle is working on using publicly available data, in a sense, publicly available knowledge base and pathways, and try to understand and reconstruct what are the steps. And in principle, it should be possible to trace back where actually dexamethasone is on what dexamethasone is acting. And then anyway, yes, it's probably, it has many targets, but yeah, we need to start from.
Simply thinking it would help if we could compare untreated and treated patients, but that will be difficult because of Peter's recovery study. Everybody's treated, obviously.
Well,
so another related question, maybe to Peter and maybe also to Cosimo, is there was a question whether we could now, based on COVID results, because Cosimo was mentioning there's a lot of commonalities between influenza and COVID, whether we could predict whether dexamethasone steroids would also have an effect on other severe respiratory infections like flu.
It's interesting. I had that discussion just yesterday about the long-term future of the platform trials, and actually the dexamethasone comparison was originally planned for pandemic flu, and so we have a question now that should we continue recovery as a platform for other pathogens like RSV and influenza, and if so, would one of the first things to test to be whether immunomodulation worked in those diseases? So I think the question is absolutely right, that the transcriptomic work could help us see whether that's a worthwhile path.
I think, another study, another PREPARE study, the REMAP-CAP study, is certainly also going to look beyond COVID at other respiratory viruses and the effect of steroids. So I think it's a valid question. I think, observational studies in flu have shown harm. So all the clinical guidelines for flu argue against giving steroids, and maybe that shows the weakness of observational studies. And again, highlighting the strength of studies like the recovery study and the REMAP-CAP study. Okay, so I have another question, this time from Malte. So I got this question whether you mentioned gene expression profile is pending. Are you going to work together with Cosimo to compare adults to children?
I mean, that's certainly the plan. It will be very interesting to see that when we will have the data ready for adults already, and this will make it possible for us to identify the most interesting patients from the pediatric cohort or from the pediatric case control design, really, to have the best possible comparison between the pediatric and the adult data. So certainly one thing that we will need to do is once we know what is associated with severity of disease in adults, we will be able to see whether this is also true for children.
Another question was posted. Well, another question to Malte was posted whether you have considered, similar to the MERMAIDS adult study, to reactivate the MERMAIDS pediatric study to enroll children, because there's a paucity of data on children with COVID. And I understand this question because children get very mild disease. So what's happening there?
So if I could have my personal choice, I think the ideal thing to do would be to reactivate the pediatric study in maybe at the end of this year or maybe in the next RE season, because what we have to realize is if we look at hospitalized children and hospitalized for acute respiratory infections, it is very, very difficult to run a study at the moment. And we are trying this with several studies in the European setting at the moment that we had started before the beginning of the pandemic, and where we've basically got zero recruitment. So it will be relatively inefficient to do the study at the moment. But it will be all the more important to see how much SARS-CoV-2 will establish itself as a pathogen in the pediatric population. So I think the ideal time to reactivate the PET MERMAIDS study would probably be towards the end of this year or even just next year.
Right.
I just say,
yeah, sorry.
We are recruiting children into recovery.
Mm-hmm. Right. But recovery, does recovery also sample, for instance, for more detailed analysis?
No, it doesn't.
Mm-hmm. There's a small question for Cosimo. You mentioned innate modules in mild and severe, and adaptive modules, or innate modules in mild and moderate, and adaptive modules in severe. And the question is this not a question of time? Is it not just because people with severe illness have a longer duration since the onset of illness?
Well, yeah, certainly that could be, but we looked into the correlation, association with time and gene expression variability. We noticed that most of the signal was due to age rather than onset of illness. But the other question is, and we are currently doing that, what happens, and I can already tell you that after we adjust our gene expression profile for time of illness, well, the pathways that emerge and the modules that emerge are pretty much the same, and the patterns of expression are also the same. So it means that even after adjustment for time, for duration of illness, we still see this behavior in gene expression. It could be that in severe cases the host is fighting, or the response is both. It's the sum of duration of illness plus some pathogen or pathophysiology specific kind of alteration and perturbation.
Thank you. So maybe there's time for one last question, which I see here on my screen, is can you use transcriptomic profiles to study vaccine responses and side effects? That's
absolutely, yes.
Top item at the moment.
Yeah, sure, absolutely. I think this has been done, with flu quite extensively, and most of the actually publicly available modules that sometimes could serve as validation set are actually retrieved from or collected from flu studies and on vaccination on flu. So yes, the short answer is that's definitely possible, and I'm really curious to look into that in the future. Yeah.
And we do collect vaccination status as part of the study, so there is a possibility to stratify for people that have been given different vaccines.
Yeah,
so it all is to be continued because there's so much, there's such a wealth of data.
Yeah.
We still need to analyze for different questions. So that's all beautiful. It's almost time, and I would like to spend the last minutes by thanking the presenters. I think the talks were really interesting, showing the importance of the MERMAID study, the wealth of data of the MERMAID study, and there's still so much to come. And I would like to thank all the people who have listened in. There are many people have listened, many people have asked questions. So I think it has been quite a lively webinar. I would still like to have a webinar, a face-to-face meeting, and I hope that will happen soon because I miss all the guys on screen very much. And I would like to invite all the people who have listened into this webinar to next week's webinar. It's on the 24th of March, next Wednesday. It will also be about a mermaid study, but this time on parvoviruses. I promise you that will be a very interesting webinar. And I thank you all and hope to see you all soon again. Thank you very much.
Thank you, Mette.
Thank you.